Hello everyone, I am Vijay and I am a PhD student from the Research School of Computer Science at the Australian National University. Today I am going to present my work GraphBin2, which is a tool that performs refined and overlapped binning of metagenomic contigs using assembly graphs. Metagenomics involves the study of various genetic material obtained directly from communities of microorganisms living in natural environments. A microbial community is usually a complex mixture of multiple species, and recovering these species is crucial to understand the behavior and functions within such communities. What is metagenomics binning? Let's have a look at this analogy. We all know about jigsaw puzzles. You get small pieces of a bigger image and you have to piece the puzzle together to form that image. Imagine you have 4 jigsaw puzzles and all the pieces are mixed together. You don't know which piece came from which puzzle. You will have to separate the pieces belonging to each puzzle and then individually solve the 4 puzzles. A similar thing is done in metagenomics analysis. Here, the mixed up puzzle pieces will be metagenomic sequences. Similar to separating the puzzle pieces, we separate the sequences into bins. These bins can represent different taxonomic groups. Then, we construct the genomes from individual bins similar to the way we construct the individual jigsaw puzzles. The step where we separate the sequences into bins is called metagenomics binning. There are 2 commonly used pipelines for metagenomics analysis. In the first pipeline, we bin reads and then assemble the reads in each bin separately. In the second pipeline, we first assemble the reads into much longer sequences called contigs and then bin these contigs. Due to the short read lengths of next-generation sequencing data, reads are usually too short to enable accurate binning results. Hence, the typical pipeline followed will be to assemble short reads into longer contigs and then bin these resulting contigs into bins representing different taxonomic groups. We can identify 2 main approaches for binning metagenomic contigs. The first approach is the reference-based approach. Here we compare the similarity of sequences with the reference database. Then the sequences are assigned to reference genomes with the highest similarity. The major problem of this approach is that it may not be applicable in many metagenomic samples when the reference genomes are not available. The second approach is the reference-free approach. Here we use the information of the sequences itself to identify the bins. In our work, we are considering reference-free approaches to perform binning. There are 2 kinds of information commonly used to support reference-free binning of contigs. First is the composition information. We obtain short strings of length k called kmers to represent the composition of contigs. Then we have coverage information where we use the coverage of contigs. We can obtain the coverage using the number of reads that align to a contig. Majority of the latest contig binning tools make use of both the composition and coverage information to perform binning. How can we obtain contigs? This is where assembly comes into play. Once we have obtained reads by sequencing a sample, the assembly process starts by identifying overlaps between reads. Then, the initial assembly graph is constructed by connecting reads with overlaps. Most existing assemblers use graphs as the underlying data structure to capture the overlaps between reads. Then, the final assembly graph is constructed by collapsing non-branching paths. These non-branching paths correspond to contigs. In metagenomics analysis, first we assemble the metagenomic reads into contigs using the assembly graph. Most contig binning tools consider only the contigs and ignore their connections in the assembly graph. Then they bin the contigs based on their coverage and composition information. Keep in mind that contigs are assembled using the assembly graph. Valuable connectivity information among contigs can be obtained from the assembly graph and used in binning. Let's see how we can address the challenges faced by existing contig binning tools. The first challenge is binning very short contigs. The majority of the existing contig binning tools bin contigs which are longer than a given length threshold. For example, MaxBin2 has a length threshold of 1000 base pairs. Contigs shorter than this threshold are discarded. This is because short contigs have unreliable composition information. Hence, many short contigs within the assembly will be discarded, resulting in low recall values. You can see that majority of the contigs in this assembly of two genomes are discarded. If we consider the connections between contigs in the assembly graph, we can recover many short contigs by considering their neighborhood information in the assembly graph. The second challenge is binning contigs shared by multiple species. Different bacterial genomes in a metagenomic sample may share similar genes and genomic regions. For example, let's consider that we have two genomes which have a shared region R and reads are obtained from them. We can assemble the reads to obtain the contigs. However, majority of the existing binning tools will assign this contig with the shared region into one bin. If we have the assembly graph with the contigs, then we can see that the contig with the shared region connects to contigs of both genome 1 and genome 2. Moreover, the contig R may have an elevated coverage value as well. We can make use of this additional information from the assembly graph and determine that contig R belongs to both bin 1 and bin 2. This is one major contribution offered from our work GraphBin2. This is the overall workflow of GraphBin2. Let's walk through each of the steps. The preprocessing steps of GraphBin2 assemble metagenomic reads into contigs using the assembly graph and then bin the contigs using existing contig binning tools. Here, vertices denote contigs and edges denote overlaps between contigs in the assembly graph. The colors represent different bins. GraphBin2 takes this labeled assembly graph as the input. Let's see what happens to this assembly graph inside GraphBin2. GraphBin2 consists of 4 main steps. Let's see what happens in each of these steps. The first step is to remove labels of unsupported vertices. Let's see how we can identify unsupported vertices. Consider the example assembly graph given. If we consider vertex 24 in blue, we can see that it is directly connected to vertex 20 in blue. It is supported by another blue vertex. Moreover, vertex 18 in green connects to vertex 8 in green through a path consisting of unlabeled vertices 19 and 14. Vertex 18 is supported by another green vertex. Hence, the vertices which have a path to other vertices of its own color are considered as supported vertices. If a vertex does not have any path to other vertices of its own color, then they are considered as unsupported. For example, we can see that vertex 1 in blue connects to only red vertices. Hence, it is unsupported. We remove the labels of such unsupported vertices. In the second step, we correct the labels of inconsistent vertices. We identify inconsistent vertices based on the labeled vertices in their neighborhoods. For example, consider vertex 18. It connects to 4 blue vertices and 3 green vertices. We can see that the 4 blue vertices are located closer than the 3 green vertices. We can see that it is more probable to push labels from the blue vertices than from the green vertices to vertex 18 as the blue vertices are located closer than the green vertices. Hence, vertex 18 in green becomes an inconsistent vertex as it is more closer to blue vertices than green vertices. Similarly, vertex 22 becomes inconsistent as well. The label of vertex 18 will be corrected to blue and the label of vertex 22 will be corrected to green. In the third step, we propagate the labels to unlabeled vertices. Unlabeled vertices can get labels from their neighbors. We have to push the labels of labeled vertices and propagate towards the unlabeled vertices. To order the label propagation process, we obtain a set of candidate propagation actions for each unlabeled vertex. A candidate propagation action for a vertex V is defined as shown. Here T is the nearest labeled vertex and we have the distance and the coverage difference between the two vertices. Contigs of the same species are close in the assembly graph and tend to have the same coverage. By this ordering, we can determine which labels to push first. GraphBin2 puts more emphasis on the connectivity information than the coverage information. This is because the edges in the assembly graph are expected to be more reliable than the coverage information on vertices, especially for vertices denoting very short contigs. Consider that we have two candidate propagation actions as given. We can push the label of vertex 6 to vertex 1 or we can push the label of vertex 18 to vertex 19. The distance is 1 in both the actions. Which action should occur first? Even though the distance is equal, the coverage difference is not the same. Vertices 1 and 6 have a coverage difference of 0, which is less than the coverage difference between vertices 18 and 19, which is 3. The more closer the coverage is, the more probable the contigs have the same label. Hence, action 1 will be performed before action 2. The animation here shows the step-by-step label propagation process for the remaining unlabeled vertices in our example assembly graph. Candidate propagation actions for each of the unlabeled vertices are recorded and are executed according to the lowest distance and the lowest coverage difference. In the last step, we infer the multi-labeled vertices. We identify possible contigs which belong to multiple species. Before moving to the method, let's see what are the characteristics of shared contigs between multiple species. These contigs connect to other contigs in those genomes and their coverage should be equal to the sum of the coverages of those genomes. Keeping these characteristics in mind, let's see how we can detect such multi-labeled vertices. First, we check all the labeled vertices that are connected to vertices of multiple different labels. In this example, vertex 3 connects to both red and green labeled vertices. Vertex 14 connects to red, green, and blue labeled vertices. Moreover, vertex 25 connects to both green and blue labeled vertices. Next, we get the coverages of each component connected to those vertices. In this example, we can roughly calculate the coverages of red, green, and blue components as shown. Consider vertex 3 with a coverage value of 108. It is in the green component and connects to two vertices in the red component. Now we have three possibilities for the membership of vertex 3. It may belong to only the green component, it may belong to only the red component, or it may belong to both the red and green components. We have to check the coverage values for each of these possibilities. If we list down the coverage values for all the possibilities, we can see that the coverage of vertex 3 is closer to the coverage of green and red components. Hence, vertex 3 belongs to both red and green components. Let's consider vertex 14 with a coverage of 19. We can see that there are 7 possibilities for the membership of this vertex as it connects to all red, green and blue components. We can see that the coverage of vertex 14 is closer to the coverage of the red component. Hence, vertex 14 only belongs to the red component. Finally, we can detect two multi-label vertices in this example, vertex 3 and vertex 25. Finally, GraphBin2 will output the final binning result with possible contigs that may belong to multiple species. We evaluated GraphBin2 using simulated and real datasets. Datasets were simulated using in silico seq tool. Each dataset was assembled using both Metaspades and SGA assemblers. We use MaxBin2 and SolidBin to get the initial binning result. In order to obtain the ground truth, we use BWA-MEM to map the contigs back to the reference genomes. If at least 50% of a contig aligns to a species, then it is considered to belong to that species. A contig is considered as single-labeled if it belongs to only one genome. A contig is considered as multi-labeled if it is longer than 1000 base pairs and it belongs to multiple genomes. Isolated contigs were not considered for the ground truth. To evaluate our method of contig binning, we applied the three common criteria: precision, recall, and F1 score, which have been used in previous binning studies. Here we have shown some representative results for MaxBin2 on the Metaspades assemblies. The results are for the 20 genome dataset and the complete Sharon dataset. MaxBin2 is one of the state-of-the-art contig binning tools available at present, but it has ignored many contigs as you can see from its recall values. GraphBin2 has been able to gain significant improvements in the binning criteria over MaxBin2, especially for recall. We will see how the assembly graph of the Sim5G dataset assembled using Metaspades undergoes refinement in GraphBin2. Figure A shows the initial labeled assembly graph obtained using MaxBin2. You can see that many contigs are not labeled. Figure B shows the assembly graph after refining unsupported and inconsistent vertices. Figure C shows the assembly graph after propagating labels. Almost all the contigs have been labeled by GraphBin2. Figure D shows the multi-label vertices marked in black. We evaluated the performance of GraphBin2 with its multi-labeled inferencing capabilities. The results shown here are based on the MaxBin2 results for the Metaspades assemblies. For the evaluation, we use the criteria sensitivity, specificity, and balance accuracy. We use the balance accuracy because the datasets are imbalanced. The number of multi-labeled contigs is much smaller than the number of single-labeled contigs. We can see that GraphBin2 has assigned correct labels for most of the multi-labeled and single-labeled vertices. However, results may get affected due to the poor performance of the initial binning tools and poor assembly quality, which can result in very fragmented assembly graphs. In the future, we plan to extend the capabilities of GraphBin2 to explore binning long-read assemblies. We have made our program freely available at GitHub for the community. I would like to acknowledge the support of Dr. Yuling and Mr. Anuradha Wickramarachchi from the ANU Computational Genomics Group. Our research was conducted using the resources at NCI Australia. Thank you very much for your time, and I'm happy to answer if you have any questions.